It is my pleasure to introduce not one, but two research scientists from Kristen Ardley's lab. It's Amrita Sule and Amy Guillaumet. Amrita holds a Bachelor in Science and a Master of Science in Life Sciences from the University of Mumbai and earned her PhD in molecular biology and biochemistry from Virginia Commonwealth University, where she studied DNA damage response mechanisms in Christopher Valerie's lab. Amrita completed her postdoctoral training at Yale University in Ranjit Bindra's lab, focusing on combination therapies targeting DNA repair pathways in solid tumors. She'll be talking first, and then following her will be Amy, who obtained her PhD in the Autonomous University of Barcelona, Spain, where she focused on epigenetic deregulation in hematological malignancies. In 2014, she started her first postdoctoral position in Holger Hein's lab, focusing on single-cell sequencing technologies. In 2017, she joined Birgit Knokel's lab at Dana-Farber Cancer Institute to study resistance in T-cell acute lymphoblastic leukemia and CAR T-cell therapies, applying the knowledge she has gained and developed in Holger Hein's lab. Whew. Okay, so let's get started. Please, again, ask questions throughout with the microphones. Thank you.
Thank you, Michelle, and thank you for having us here today. This is our very first time at the MPG Primer, and we are very excited to tell you guys all about the Developmental GTEx project. Before I move on, I just want to thank all the donor families who have generously gifted us such precious gifts to support the important work. And just to note that some of the slides will have histology and tissue images. Okay, so before I move on to Developmental GTEx project, I'm just going to give you a quick overview of the GTEx project, which probably most of you are aware of. So the GTEx project, which was launched in 2010, aimed to map the effects of genetic variation on gene expression on a broad range of tissues. The primary goal was to improve the understanding of regulatory mechanisms in complex traits and diseases. GTEx collected over 50 different types of tissues in around 1,000 postmortem adult donors. And they also developed protocols which were very standard operating protocols for tissue collection, processing, as well as facilitated open data sharing via the GTEx portal. The gene expression and the splicing data was mostly generated using bulk RNA sequencing. Through these tools, we also identified cis and trans eQTLs. Altogether, GTEx has been pretty instrumental in linking genetic variation to regulatory function, and thus helping researchers in to connect non-coding variants, which make up most of the GWAS hits. And this helps us to understand biological mechanisms and disease pathways. So all of the GTEx work, for most part, generated whole genome sequencing data and bulk RNA sequencing data. However, with recent advances, we have tools like single-cell RNA sequencing available to us now. Technologies like these have enabled profiling gene expression at a resolution of individual cells, offering much deeper insights compared to the bulk RNA-seq. Majority of efforts like Human Cell Atlas, PsycEncode, HubMap, BiCAN, and more are leveraging these tools to systematically map transcriptomic profiles across human tissues at cellular level. Single-cell RNA-seq, as we all know, allows us to resolve cell types, states, and even differentiation trajectories within tissues. These efforts by these consortiums, however, are very important. However, most of these focus on adult tissues, and most do not capture developmental trajectories. There has been some effort by the Human Developmental Cell Atlas, which is beginning to fill these gaps. They focus mainly on prenatal tissues. However, these studies are generally focused on sampling the same tissue across multiple individuals as opposed to multiple tissues from the same donor, which hampers cross-tissue comparisons and investigations into effects of genetic variation on gene expression. So there is a need for pediatric normal reference data. As we know, a comprehensive understanding of normal human development is critical for uncovering origins of many diseases. A number of inherited disorders from congenital malformations, heart defects, neurodevelopmental disorders originate during gestation. Additionally, there are pediatric cancers which are believed to arise from transient development cell types that are not captured in adult data sets. Also, both genetic and environmental influences during early development can predispose individuals to diseases that manifest later in life. All this underscores the need for pediatric-specific reference data sets to accurately study disease mechanisms and identify their developmental origins. So here I would like to introduce the Developmental GTEx project, which was designed to extend the foundational GTEx resource by incorporating a developmental dimension. While GTEx focused on adult tissues, DGTEx aims to profile gene expression across key stages of early human development. This project will collect tissues from 120 postmortem donors stratified into 4 developmental age groups: infancy, early childhood, prepubertal, and postpubertal. Each group will include approximately 30 donors, enabling cross-sectional analysis of gene expression changes across developmental stages and across tissues. This data will support investigation into age-specific gene regulation, developmental trajectories, as well as early origins of disease. Now, in parallel to the human collection, DGTEx includes a non-human primate arm to enable cross-species comparison of developmental gene expression. We are sampling 126 rhesus macaques, 72 marmosets across a range of developmental stages covering both prenatal and postnatal periods. The postnatal time points are aligned across species between humans, marmosets, and macaques, which allows comparative developmental analysis. Importantly, the prenatal non-human primate samples provide rare and ethically feasible access to developmental time points, which are not accessible easily in humans. This design enables to better understand conserved and species-specific aspects of primate development, especially during early life stages. Now, there is still a limited understanding of how well regulatory networks and developmental programs are conserved between humans and non-human primates. Most of what we know about human biology, both in normal and disease state, has been studied in model organisms like rodents, especially mice. However, mice are evolutionarily distant from humans, especially they exhibit key developmental differences during early postnatal stages. These differences limit their translational relevance of findings. As we have seen in some of the clinical trials which failed despite having strong preclinical data in rodent models. In contrast, NHPs share more similar developmental trajectories in humans, making them critical reference for understanding early human development and improving disease modeling. Therefore, establishing normal reference data in NHPs will enhance our ability to interpret human developmental data and bridge gaps between model systems and clinical research. The NHP component of the DGTEx project includes 2 key primate models: the rhesus macaque, which is also known as the old world monkey, and the common marmoset, which is known as the new world monkey. Marmosets have been widely used in biomedical research due to their small size, high reproductive efficiency, and their tendency to produce dizygotic twins sharing a placenta, a feature valuable for developmental studies. Rhesus macaques are the most extensively used NHPs in research and are phylogenetically very close to humans, especially comparable uterine and placental structures. Together, these 2 species offer complementary perspectives on primate development and help model the conserved and divergent aspects of human biology. Taken together to summarize, the DGTEx project aims to build a comprehensive reference atlas of normal tissues across 3 species. Here we are again, just to recapture, we are profiling a variety of tissues from 120 pediatric donors across 4 age groups, and also rhesus macaques and common marmosets. The ultimate goal is to define developmental and tissue-specific molecular patterns across these 3 species for integrative analysis using a number of sophisticated tools like bulk RNA-seq, single-cell RNA-seq, whole genome sequencing to help characterize gene expression, chromatin accessibility, and gene variation. I'll let Amy continue.
Yes, thank you so much, Amrita, for the wonderful introduction to the Developmental GTEx project. Building upon that, I'm going to dive into how we actually acquire the critical human samples for the project. Our human sample collection relies on the network of the organ procurement organizations, or OPOs, across the United States. We partner with 6 of them, including Connect Life and Life Gift, amongst others. These OPOs provide us with significant amount of volume of pediatric donors referrals to screen for eligible donors. Now, once a potential donor is identified by an OPO, the next critical step is to determine donor eligibility. This process is very rigorous and relies on specific criteria. We don't reinvent the wheel here. We actually leverage the existing organ and tissue transplant donor screening processes that the OPOs already have in place. This structured approach provides donor families the opportunity to consider donation for the Developmental GTEx project. As you can see on the box on the right, this outlines some of the key inclusion and exclusion criteria that guide our selection. Donors have to be 18 or younger. They have not received blood transfusion within the 48 hours, amongst others. We also are collecting a large amount of donor metadata. This builds directly of best practices from the original GTEx project. The de-identified donor level data includes critical information such as demographic details, comprehensive medical history, birth circumstances, environmental exposure, amongst others. So now, once a donor is eligible and metadata is collected, the crucial step is the tissue sampling itself. First, the Developmental GTEx tissue sampling utilizes common coordinate framework. This is critical because it enables reproducible sampling across diverse sites, different donors, and even different species. This ensures that a sample taken from, say, a specific region of the lung in one donor is comparable to a sample from the same region in another developmental stage of another donor. Secondly, the collected tissue sites and all the metadata gathered are aligned with other consortiums, as Amrita mentioned before. So sampling is performed by highly trained recovering teams with introduction of diagrams and images as visual guides for tissue procurement process. To ensure the highest quality, on-site instruction by dedicated pediatric pathologists is consistently available at the OPOs throughout the project. Now, let's look at the specific sampling schema itself and how it varies is based on the unique size and structure. So in general, most tissue sites, we aim to collect a small standardized samples. For solid organs, we collect a 2x5 by 1 by 1 cube. And for mucosal organs, we'll collect a 2x5 by 1 centimeter in its full thickness of the organ. Each of these samples is divided into 3 oligos, as you can see in the image. 2 of them will be frozen for molecular analysis, and 1 will be fixed for histopathological evaluation. One key important aspect and beauty of this project is that we will maintain spatial orientation of these tissues. So the frozen part is adjacent to the fixed part, and the fixed part is adjacent to the other frozen part. So we know specifically the orientation of these tissues. Okay, so however, studying developmental introduces a unique challenge. Organ size changes significantly during development. As you can see here on the left, heart sampling schema changes from birth to adulthood, where we'll be collecting whole hearts in non-human primates. And then for younger donors, we'll be collecting sections, and then for older donors, we'll be doing subsections. Again, this is another example of a testis sample where younger donors will be collecting the whole organ, young donors will collect the full organ, but older donors, the samples will be sections. Understanding and adapting to these developmental changes in organ size and structure is vital to ensure that our samples are truly comparable across the entire pediatric age range, allowing us to accurately track developmental and gene expression patterns. While we cover the human side of collection in detail, it's crucial to also highlight the tissue sampling in our non-human primate collection. Tissue sampling for the non-human human primate is designed to map the human tissue sampling as much as possible, with modifications from species-specific anatomical and developmental differences. The non-human primate won't be affected by organ donation, so we'll have additional tissues. Tissue ischemia is minimal because as soon as sample collection begins immediately from euthanasia. We also have, as Amrita mentioned before, we'll also be collecting prenatal samples. Okay, So now, having discussed eligibility and sampling protocols, I want to highlight the impressive scale and depth of tissues that we're actually collecting in the Developmental GTEx project. For the human donors, on average, we're collecting 20 different tissues directly on site, and then there will be secondary dissections, up to 72 different tissue from a single donor. So similar for the non-human primates, we're recovering up to 50 to 60 tissues, and then we'll have secondary dissections that will go up to 80 distinct tissues per animal. As you can see here, we'll be collecting the skin. There's a comprehensive list. The brain, the thyroid, the pancreas. So yes. So to maximize, certain organs will be recovered in whole, like the hearts, the lungs, and the brains. And these will be sent out to specific places where the heart will be sent out to the children to chop, where it'll be further subdissected. The lungs will be sent to the University of Rochester for subdissection, followed by the lung MAC protocols, and the brain will be sent to the University of Maryland, and then further subdissection to the University of Yale. Okay, so I want to take a step back and provide a high-level overview of the entire Developmental GTEx workflow. This schematic details the comprehensive journey of our samples from donor to data to ultimately to discovery. As you can see here, the workflow presented in two parallel streams. The left side outlines the process for our human postmortem donors, where you can see we'll be collecting 120 donors. All of our samples will have pathology review. Then the whole full organs will be sent to us to the broad, will be biobanked, and on the other side we'll have the non-human primates, the marmoset collection from MIT, and the macaques from Oregon will be sent to us at the broad, where we'll biobank them. So yes, Amrita.
Thanks, Amy. As Amy provided,
Can I ask a quick question?
yes.
We have an anonymous attendee online asking about if the cause of death, like kidney failure or cancer, the person, will this affect the results, or are they excluded?
For donation, yeah.
There's a whole list of eligibility criteria. Yeah. Thank you. Yes, so as Amy described the workflow when a donor is authorized and what kind of samples are collected, there are some samples which are collected fresh and they have to be processed as soon as possible. One of them is blood. Till date, we have collected around 44 diseased pediatric donors. And for each donor, we obtain two types of blood samples. Frozen blood, which is frozen on site and is usually primarily used for DNA extraction, which then downstream can be used for whole genome sequencing. We also collect fresh blood. This blood is shipped to the broad for downstream processing. From this, we isolate leukocytes, which are then used for single-cell RNA sequencing to profile gene expression. Now, it's very important to evaluate sample quality, or we need to consider sample quality, and this is dependent on three key time points. One of them being, when is the blood collected? What is the time from the passing of the donor to the start of the collection that has passed? Then time from sample collection to shipment initiation, and time from shipment to receipt and processing of the blood. All these time variables are crucial because delay in any of this can actually hamper downstream RNA integrity and thus affect downstream data quality. Faster turnaround helps preserve transcriptomic data fidelity as well. This is at the bottom an example of a donor. Here you can see that 0 hour marks the time of passing. Then after 20 hours, the blood was collected. 7 hours after that, the shipment was set up. And by the time the blood reached broad, 35 hours, 47 minutes had passed. This changes from donor to donor, but this is just an average time what it can take. We receive blood at broad after hours on weekends, and the broad security assists us. We can track the shipment, and then somebody from the lab retrieves the blood and processes it so that we can make sure we take care of it as soon as possible. Now, when we actually, so our goal is that from the fresh blood, we can isolate white blood cells, which are also known as leukocytes. These are critical component of the immune system and help body basically fight infections, inflammation, and disease. Although they make a small fraction of the whole blood, they do have distinct cell types like T cells, B cells, monocytes, NK cells, and all your granulocytes. They provide basically a snapshot of the immune activity in the body of the donor at the time of sampling. In pediatric donors, especially, it can help us to understand how immune system develops, responds to stimuli, or maybe how it may be altered in a disease. Now, after we receive fresh blood samples in the lab, our first step is to get rid of all the red blood cells. We do this by a magnetic separation. This helps us to enrich WBCs and also get rid of all the noise that could come from RBCs. Once RBCs are depleted, the enriched WBC suspension is processed through this honeycomb hive capture assembly. These are basically these contraptions you see on the left bottom corner, where each of these contraptions has wells in which individual white blood cells are captured, and then they each receive a unique molecular barcode. Once the cells are captured, they can be stored for a long time in the -80, and we can then basically process them in batches. We don't have to process the downstream single-cell RNA sequencing per donor. We can have them as a batch and then process them. One good thing about this hive assay is that it also helps us preserve granulocytes, which are often lost by other traditional methods. We also do PBMC isolation and storage for the non-human primate species, which are done. So the marmosets are done at Broad, and the macaques' blood is processed in Oregon ONPRs. In addition to blood, we are also collecting skin tissue samples from donors to isolate fibroblasts. So when we receive a skin sample, we basically, and this also is received at the same time as blood. So once we receive the skin, we start by carefully trimming the skin sample, removing the fat, and then this tissue is then cut into small pieces and allowed to adhere on a petri dish on a gel matrix. You do regular cell culture, change media, do like a single passage so that you obtain primary culture. And once at a correct confluency, we can cryopreserve these fibroblasts, and these are biobanked in GP. Now, these fibroblast cultures basically allow us to use in downstream functional assays. We can reprogram them to iPSCs. Also, these are important resources that reflect the living cells. They basically reflect the donor's genetic and environmental history in form of living cells. Another thing Amy mentioned is that all the tissues what we collect they undergo pathological review. So we collect a frozen tissue, and then we have a fixed tissue, another frozen one. So the fixed tissue, all the fixed tissue samples undergo detailed review by a pathologist. The review serves multiple critical functions, from initial quality control, tissue identity, as well as quality assessment. So in the initial quality control, the pathologist verifies that the labeled tissue matches what's actually on the slide. Misidentification can compromise downstream data analysis. So the tissue mismatch is verified. They also check for orientation issues if the tissue was embedded or sectioned in a way that might limit interpretability. Then it's a technical slide quality issues. Just to overall look at the quality of histology slide, including staining quality, tissue folding, section thickness, etc. Once the initial QC is complete, the pathologist evaluates the biological quality of the tissue. Each tissue gets an autolysis score. This indicates how much postmortem degradation has occurred. High autolysis can obscure tissue architecture and limit utility. Autolysis score is usually given from a scale of 0 to 3, 0 being good and 3 being bad. The pathologist also looks for the presence of target versus non-target tissues to make sure that the slide actually has the intended organ or a structure. And sometimes an unintended tissue could occur due to collection error or anatomic proximity. They also look for signs of any diseases, inflammation, or abnormal development, which was not known prior to collection. And last but not the least, maturation assessment. For pediatric samples in particular, evaluating developmental stages can provide important context for interpretation. After all these assessments, the pathological final review determination is performed, and then we decide if this tissue is suitable for downstream applications. Now, the different tissues are reviewed by pathologists at different centers. The University of Maryland Brain Bank tissue reviews scans from NHP and non-human brains. All the non-brain human tissues are reviewed at CHOP, and the non-brain NHP tissues are reviewed at ONPRC, Oregon. I'm just going to show you this one example as to what information we can gather from histology slides across development. So in the top panel, you can see that the slides show the developmental progression of glomeruli in macaque kidney, spanning from gestational age 50 to an adult. In GD50, you can see that the kidney is in early developmental stage. There are no mature glomeruli yet, but renal vesicles and comma-shaped bodies are starting to form. At GD100, glomerulogenesis is actively underway. We can identify some S-shaped bodies. GD150, which is a late gestational stage for macaque, glomeruli become more defined. And as we move from neonate, infant, juvenile to adult, we can see that glomeruli are fully mature and functionally stable. These slides will help us visualize and understand developing organs and their highly organized and fully functional aspects across ages. So we are basically cataloging this across a range of tissues in NHPs and humans. And this is going to be a very important aspect of information which will eventually be available to the community.
Amrita, so building upon that, we're now going to transition to the diverse types of data that will be generated from the Developmental GTEx project. As mentioned earlier, all tissues are biobanked and every collected tissue undergoes a detailed histology imaging. So we will be generating whole genome sequencing, bulk RNA sequencing, long read sequencing, single cell technology, such as the multiome where you obtain the gene expression attack, as well as methylation assays, and various special single cell sequencing techniques. So all of our donors will receive a whole genome sequencing to provide a complete genetic blueprint. This will allow to identify inherent genetic variation, improve genome and isoform structure annotation, and enable genetic demultiplexing as samples are batched together. So we will also be performing bulk RNA-seq on the majorities of tissues collected. So far, we have sequenced to date 264 samples from 31 donors and 46 different tissues in humans, 262 samples from 27 donors and 61 tissues in macaques, and 237 samples from 18 donors and 59 tissues in marmosets. So just a quick overview of the PCA plot and T-SNE plot, where we obtain a separation of the different tissues, indicating the high quality across the data and the species. So another assay that we're going to perform, it will be full-length RNA sequencing using mass iso-seq to enhance our understanding of isoforms throughout development. This method was developed here at the Broad by Aziz, and we've already performed a pilot study where we took 12 humans, 6 marmosets, and macaques, different tissues in different age ranges. And as you can see in the heat map, we already have tissue type that drives primary clustering and a secondary clustering driven by species. So there's a lot of biology to be discovered here. So another core assay that we'll be doing is single cell sequencing, where we'll be profiling from the same single cell the gene expression and the chromatin accessibility using 10x genomics. So other assays, gene expression and attack, the 10x multiome will be our core assay, but we will also be doing single cell methylation and spatial transcriptome on some of them. So these powerful technologies move us beyond bulk averages, providing unprecedented precision to map development at the cellular level. So just to elaborate on our core high resolution assay, this slide provides a brief overview of the 10x genomics multiome, a technology central to the developmental GTEx, where we'll be linking gene expression and chromatin accessibility from the same nuclei. So briefly, for those who don't know the technology, you take frozen tissue, you isolate the nuclei. Each tissue requires a specific protocol for isolation. Once you obtain the nuclei, you perform the 10x multiome, which is by doing droplets of single cell. You will then perform the libraries, attack gene expression, and you will obtain the gene expression and the chromatin accessibility of that nuclei. So as mentioned earlier, so this will be our main core production, but we are in parallel doing the same experiments at Yale University. So as Amrita mentioned before, the tissue that we will be doing performing single cell sequencing has to be of high quality data. So we will select tissues that have a high RQS score and that have a good autolysis score. And then on the lab side, validation of for good data will be intact nuclei and the traces we obtain from those libraries. So we will be generating a beautiful multi-omi data set where we'll be having the 3 species. We'll select 2 age ranges, the young and the older side, and where we have matched the tissue types. So we will be generating 10x multi-omi data on all of these brain regions, amygdala, thalamus, hypothalamus, cerebellum, and kidney, pituitary, muscle, quadriceps, lung, liver, thyroid, muscle, and adrenal gland. This is going to be a beautiful data set where we'll see a lot of biology within tissues and within species. Here's just a glimpse of a data set that we've generated. So this is a kidney sample where we did 10x multi-omi, where you get the gene expression and the attack. Here you can see beautiful clustering, both in the gene expression and the attack, where we get high number of genes per cell, high number of cells. And for the attack metrics, we get good TSS score. So this is human kidney and high fragmentation, high quality, overlapping peaks. So this is just a glimpse of the kind of data we'll be getting from the 10x genomics multi-omi. So as mentioned before, another workflow that we'll be performing will be methylation profiling at the single cell level using scale biotechnologies. So again, briefly, you isolate the nuclei, you fix them, then you perform combinatorial indexing, isoform sequencing. Again, you barcode them, you sequence them, and then you analyze them. So we performed a pilot data set on kidney where we got 2 younger donors and 2 older donors, and we performed this assay. And we're still looking into the data, but you can already see beautiful biology where there's a specific infant cluster that is specific only to the younger donors. So there's a lot to be discovered in these data sets. We're really looking forward to it. And lastly, we'll be performing spatial assays to understand tissue organization and cell interactions. For the spatial assays, we have tried slide tags on kidney, again on the same, on human kidney. So briefly, with this technology, you place a section of tissue on the sequence spatial barcoded array. You tag your nuclei with photocleaved spatial barcodes. You isolate the nuclei and you perform 10x genomics, where you'll capture the mRNA and the spatial barcodes. With this pilot, as we took the kidney, we placed one of these arrays on the medulla and on the cortex. And as you can see here, we have a Disney plot on the medulla and on the cortex, where you can see the specific different subtypes from the different anatomical region. Again, a lot of biology to be discovered. So what are the outcomes of this big project? So tissue collection and data generation are currently ongoing. Data and pipelines will be available on the GTEx portal. On the portal, you'll be able to select the data for interactive visualizations, and all tissues will be biobanked for future reference, for future research purposes. And to conclude, to bring it all together, the Developmental GTEx project represents an unparalleled effort to transform our understanding of pediatric development and its evolutionary foundations. It is crucial to remember that pediatric tissues are fundamentally different from adult tissues. They have uniquely cell type compositions and distinct physiology. To study the evolutionary conservation and differences of developmental programs between humans and non-human primates. And the Developmental GTEx project will provide a groundwork and infrastructure for many research studies. For example, elucidating the origins of pediatric diseases, childhood cancers, to developmental disorders. Everything that we presented here today we wrote a marker paper that describes all of the project. If you're interested in learning more about it, what tissues will be subsampling, the exclusion criteria that was asked before, that's also in the paper. That you can find online. So the most important, I want to acknowledge, and we're extremely grateful to the Developmental GTEx donors' families who have generously provided such precious gifts to support our research work, as well as all of the non-human primate collection. And the Developmental GTEx Consortium, it is amazing to work with such a big team, and all of our Broad colleagues here at the Broad and our industry collaborators. So yes, thank you very much.
Do you have any questions?
Two questions. One, I'm curious about how recruitment is going. Is that being a bottleneck? And are you getting to be picky about maybe like having some other dimensions of diversity for your age and your data set? And the other is, I'm just feeling so aware that this is 120 dead children, many of whom are dead.
Yeah.
And those families, and I'm wondering, is there a plan for kind of ongoing relationships with those families, giving you updates about the project?
Yeah, thank you for the question. So I'll answer the second question first. So we actually have an LC initiative where basically they connect back to the families and they are informing them how their gifts are, what are we trying to do with them. So there's a constant communication aspect. So there's actually also a LC paper, which they're going to publish soon, which talks about these efforts. And your first question was, how is the recruitment going? So I think it's very difficult because also getting consent because of the age of donors, it's not very easy. So sometimes for a couple of months, there is a quiet where we don't see, but sometimes we do see a lot of donors in a month. So it's up and down, yeah. And as you said, it's 120 donors, pediatric, it's hard, yeah. But it's very precious and we do appreciate everything we get.
We have a question online from Ben Weisberg. I'll ask them one at a time because I know it's hard to deal when there's multi-part questions. Will the DNA be sequenced with long reads? You know.
But I think it all depends on and what biological interest. As we get more data, I think we'll have more questions. So.
And then another question Ben asks is, when do you anticipate the first release of data to the portal?
It's still, do you know?
I mean,
right, because it's when the 10x single cell data, and then we have the RNA ready to release. So I think
the plan is, so right now it's going through thorough QC. We have generated a lot of data. A bunch of our computational people in the team are doing thorough QC. And then the hope is to release it sometime this year. I don't want to say next month, but it's this year. Yeah.
And then the last question from Ben is, if they already have their own access to just GTEx, will they automatically have access to DGTEx? Or is there a separate application process to access the data?
We'll defer that to our PI, Kristen.
Yes.
Are there any other questions? We don't have any other ones online yet. Come to the mic.
So I was wondering, you know how you said that you have fresh blood too? And we also say that similar to SMART and GTEx, I think these ones, they don't have pre-existing disease mostly. So, and the time like to get the sample in 30 hours to Broad. So I know it's a very hard situation when that thing happened. I was wondering if the families, they register even at the time that the kid is born, like how can they say that this is going to happen if there is no pre-existing issue to deliver the sample that quickly?
So you're saying that how do we know if there are any pre-existing conditions in the blood?
Yeah, because I think I saw in one of the slides that it takes about 30 hours to get the sample.
Yeah,
and then
so we do look at viability and of the blood, and then there are certain, like for example, the Hive assays, they recommend, the viability should be say 75 to 80%. Sometimes it's 60. We'll still proceed because it's such a precious sample. And a whole range of serology is done to all these samples. So we know if there are any, like they're screened for infectious diseases and everything. So but we have some information on that when we receive samples. Does that somehow answer?
Yeah, and the second, do we have any other question or should I? So, so the frozen tissues, they're gonna degrade, right? And I think they degrade it, or no?
So no, there's a process put in place when there's the donation, tissue is collected and they follow, everything is like on frozen trays, that you don't lose a change of cold. So everything is like when they send them, there's in dry ice, when they come here to Bro, they're put in -80, so there's no degradation. I mean, you do have a lag between starting collection, but that's why we're doing all of the pathology to make sure that the assays that we do is highly good quality data. So there is a control.
And the ischemic time is set to be not exceed 24 hours. Yeah, so that is already in place. And only the fresh blood is shipped on ice. The tissues get frozen right there. So you can hope that the RNA degradation stops there.
Great, thank you. Impressive work.
Rajesh.
Thank you.
I've got a question. Thinking about de novo mutations and somatic mutations, do you have the ability to genotype the parents of the child? I might have missed it in the beginning.
That's a great question. Yeah, no plans yet, but to be seen. Yeah.
I can ask another question, then people probably want to get back to their stuff. I was really impressed about the multiome being the core assay rather than just single nucleus RNA-seq. Is there plans to, I guess I was wondering also about the idea to do both bulk and single cell. How did you pick how much you're going to spend to bulk versus single cell, or why not just put all your money in the single cell pot?
Varies.
You need a lot of money.
It's an expensive assay. So bulk is affordable. So they want to do on all the tissues, the bulk. And then we'll do this pilot, and then we'll focus on the biological questions to continue on with the single cell assays. But yes, if we could do all the tissues and all, that would be amazing to do single cell. But we're doing quite a bit now. Yeah.
Yeah. It's a balance between a good design versus how much the expenses. Yeah.
And just out of curiosity, what is the total budget? I might have missed it at the beginning.
What is
The total budget of,
Yeah.
I think you can get on the NIH, it's publicly available.
Yeah, because it involves a whole range of things from donor, like authorization, collection. So
Yeah, it's in the NIH website.
I
It's
see, yeah.
publicly available.
I see. So you don't know what it is.
Did I know? No, I don't know.
Okay,
Yeah. I can look it up later, yeah, but I don't know. No.
Any other remaining questions? We do have a little bit of time, but I know people like to enjoy their breakfast. All right.
I just
Let's-
want to say thank you to
Oh.
Rachel, who's in the audience, because she showed us the fibroblast culture. And yeah, so thank you. I just, yeah.
Yes.
Yeah, so I mean, it's been great collaborating with everybody at Broad, because when we started the project, there was a lot of things which we were learning. So people have been extremely helpful.
The Broad community is amazing, so thank you for
Yeah.
everybody.
Yeah.
It was amazing.
Yes.
Wonderful. And on that very high note, let's thank our speakers again.